Hello everyone, Mrs. Page here. So welcome to your third biology lesson. So we are looking at key concepts of biology, which we've been looking at for a couple of lessons now. So last lesson we looked at enzymes and how enzymes worked and factors that affect enzyme-controlled reactions. So we're going to do a little bit of revision of that, and then we're going to move on to look at how enzymes digest food inside our body. So what you'll need for this lesson is a pen or a pencil and a piece of paper or a notepad. So if you don't have either of those things, then just pause the video and go and grab them now. Brilliant, well done. So what I'd like you to do is write out the learning question: which enzymes digest my food? And I would like you to draw and label this diagram. If you don't want to draw it, you can just write out A, B, A to D, and then write out the labels for it. So if you pause the video and have a go at that now. Excellent, well done. So A was a substrate molecule. So that's the thing that's being broken down in the reaction. B was the enzyme. C is pointing to the active site. Remember, that's the part of the enzyme that's got a specific shape, a complementary shape that the substrate can fit into. And D is an enzyme-substrate complex. That's the term that we use in science for when the enzyme and the substrate have joined together. We call it an enzyme-substrate complex. And if you had to go at the purple question, which is what is the key word that we use to describe the ideal conditions that an enzyme works best at? It's the optimum conditions, okay? Or if you're talking about temperature, be the optimum temperature, the temperature that enzymes work best at. Well done. So what I would like you to do now is write out this paragraph and see if you can put in the correct keywords from the bottom. And then what we'll do is we'll read through it together in a minute or two. Okay, so if you want to pause the video, have a go at writing that out, please, popping in the correct keywords. Brilliant, well done. So tick the words that you got right and correct any if you need to. Okay, so enzymes are proteins. Okay, so enzymes are protein. So if you need to correct any of these, you can just pause the video at any point just to correct your answers. Okay, so enzymes are protein. They have an active site, which is a particular shape. Remember that complementary shape. A specific substrate, and that's the thing that's doing the reacting, so the thing that's being broken down or the thing that's being made. So a specific substrate fits into this like a key in a lock. Enzymes work best at an optimum temperature and an optimum pH. Okay, so optimum is just the conditions that they work best at, that there's the most enzyme activity. Fantastic, well done. So just a reminder of what the word denatured means. So if an enzyme is denatured, its structure and its function is altered. And that's because the shape of the active site has changed. This can be caused by very high temperatures or extremes of pH. So that means very acidic or very alkali conditions. Okay. So would you be able to pause the video, write that definition of denatured enzymes down? That would be brilliant. Fantastic, well done. So we looked at graphs showing enzyme activity in the previous lesson. So you can always go back and have another watch of that lesson if you need to. But remember, we can plot the activity of an enzyme on a graph under different conditions to determine where it's working the best. So what I would like you to do is match the labels on this diagram, so the letters on this diagram, match them to the correct, oh, hang on a second, sorry, it's just my computer messing around, match them to the correct sentences, so the correct descriptions of what's happening in the graph. Okay, so if you pause the video, write out A, B and C and try and write out the correct description of what's happening in the graph there. Fantastic, well done. So A, this part here, is description number 2. So heat energy causes more collisions between the enzyme and the substrate, because remember, they're moving around faster, so they're more likely to collide and form an enzyme substrate complex, and so activity increases. Okay, and B, so part B up here, part B is 3. There's an optimum temperature, so that's where enzymes are working best. Okay, so that's that high point up here. And C is number 1. So the enzyme has denatured, so the active site has changed shape and the rate falls really rapidly. The rate of enzyme activity or the rate of reaction falls really, really rapidly. Okay, well done. So if you give yourself some ticks if you got those right. So a couple of practice questions here for you to have a go at. Okay, and then that will be our revision part finished. Okay, so if you can read through these questions and try and answer them. All right, give yourself about 5 minutes to do that. You pause the video and answer these questions. Excellent, well done. So the 3 main factors that affect enzyme activity. So we looked at those last lesson. They are pH, so how acidic or alkali the conditions are, temperature, and substrate concentration. Number 2, the substance that is changed during a reaction. That's the substrate, so that's the thing that's doing the reaction. Number 3, which letter in the graph shows the optimum point? It's point B. So this part where the enzyme activity is highest or the rate of reaction is highest. Number 4, which word describes what has happened to the enzyme at point X? What's happened to it here? It has denatured, so the active site has changed shape there because the temperature is too high. And therefore the substrate can no longer fit into the active site, and so therefore the enzyme doesn't work anymore, and we call it denatured. And estimate the temperature of the optimum point. So using the graph, reading down, I would say that that's about 37 or 38. So here we go, any answer between 37 and 41, and you can give yourself a big tick. All right, well done. So mark your answers and correct them if you need to. Brilliant. So we are going to be looking at chemical digestion. So that's how enzymes help to digest our food. So there's a difference between what we call mechanical digestion and chemical digestion. So mechanical digestion is anything physical that's helping us digest our food. So I wouldn't be able to digest this steak without chewing it first, without doing some kind of mechanical digestion. So chewing it with my teeth would be that mechanical digestion, trying to break it down physically. All right. But we also carry out what we call chemical digestion, and that uses enzymes to break down the molecules in our food. So what we have are large molecules. Now, all of these large molecules look different, all right? So carbohydrates look different to proteins, and they look different to lipids or fats. Okay, so we've got our different large molecules that we've eaten here. They get broken down by enzymes into much smaller molecules. Okay. When there are those much smaller molecules, they are able to diffuse into our bloodstream, and then they can go in our bloodstream to all the cells where they're needed. Okay. So we are going to look at how these large molecules get broken down and which enzymes break down which molecules of food. So there are lots and lots of enzymes in our body, but these are three of the main ones. We've got lipase, amylase, and protease. So what do you notice about all of these words? Can you notice anything that's the same about all of those words? Okay, hopefully you notice that they all have this ase sound on the end. Now, all enzymes have got this ase, we call it a suffix, don't we? A part of a word that goes on the end. So all of them have got that ase sound on the end. So lipase, amylase, and protease. And enzymes can break down long chains of molecules like fats or lipids, carbohydrates such as starch and protein. They can break all of those very, very large molecules down into much smaller molecules. So I wonder if you can have a go at matching which enzyme breaks down which large molecule, which substance of food. So look at the start of the word. So lipase, the beginning part of lipase sounds a little bit like lipids. So lipase is the enzyme that breaks down fats or lipids. The other easier one is protease. So looking at the beginning of the word, that looks like the beginning of the word protein. Protease is the enzyme that breaks down protein molecules. And then we've got amylase, which isn't quite so easy, but amylase breaks down carbohydrates or starch. Amylase breaks down starch. Okay, now I've got a super difficult task for you. Well, I'm sure you'll find it fine. If I had this task, I would totally fail. Okay, what I would like you to try and do is best you can. Don't worry about it. It really doesn't need to look exact. You just need to have the main parts of the digestive system. So if you can pause the video and just have a go at just sketching this, like I said, it doesn't need to be accurate at all. We're only going to use it to learn where the different enzymes work. All right. So if you pause the video now, have a go at sketching out and labeling the digestive system. Well done. I'm sure you did a much better job than I could ever do. What I would like you to do now is use this table about the enzymes that we've just learnt. I'd like you to use this table to make some notes on your diagram. All right. So, what is the reaction that is catalyzed? So, what reaction is happening? So, what's the breakdown? So, starch turns into simple sugars that can be absorbed. Proteins turn into amino acids. They're just the building blocks of proteins. And lipids turn into fatty acids and glycerol. Okay, so you can write that on next to your diagram. And whereabouts in the body is it produced? So amylase is produced in the salivary glands, in the pancreas, and in the small intestine. Protease is produced in the stomach, in the pancreas and the small intestine. Because protease is produced in the stomach, it works at a really, really low pH. So protease is really interesting. It can work at a pH of 2 or 3 because the stomach is so acidic. And lipase, it's produced in the pancreas and the small intestine. Okay, so if you can make those notes from that table, if you can put those in or around your diagram, that would be fantastic. Well done. So you've listened really well, and I'm sure you've got lots and lots of brilliant information in your diagram there, and lots of information about the enzymes and what they break down. So what I'd like to do now is to just do a quick quiz to see how much we've learned. So if you could write questions 1 to 5 on your page, and then if you could pause the video and try and answer these questions, that would be fantastic. So number 1, what enzyme breaks down fat or lipids? Number 2, where is protein broken down? Remember the really low pH that protease works at. Number 3, what does protein break down into? What are those building blocks of proteins, those smaller molecules that protein breaks down into? Number 4, what happens to enzymes at high temperatures? And number 5, what do we call the molecule when an enzyme is bound to a substrate? So when an enzyme and a substrate join together, what do we call that? So if you pause the video now and write out those, and then we'll go through and mark them together. Fantastic, well done. So the enzyme that breaks down fats or lipids is called, well, that should be lipase. That's a mistake there, should be lipase. Because remember, all enzymes have got that A's sound at the end. So there's one I put in just to trick you, to see if you remember that A's sound at the end of any enzyme. Where is protein broken down? That's the stomach. What does protein break it down into? Breaks down into amino acids, those tiny molecules that are the building blocks of proteins. What happens to enzymes at high temperatures? They denature. So remember, that means the shape of their active site changes so that the substrate can't fit into them anymore, so the reaction can't happen. And number 5, what do we call the molecule when an enzyme is bound to a substrate? We call that an enzyme substrate complex. There's that really good key term that you need to know. Excellent. And that is the end of today's lesson. On Shobi, there is still a question about enzymes from last week, about the factors that affect enzyme activity. So if you haven't answered that, then go onto Shobi. The class code is on class chart, so go on to Shobi and submit your answer for that. Okay, well done for listening. Well, if you answered the questions, that's fantastic, well done. And I will see you soon for another biology lesson.